Yep. So yeah, good evening, everyone. My name is Harkirat. I'm a PhD candidate in biomedical engineering. I'm going to present sort of my journey on this project. A mechanistic neural network model optimizes both the potency and the toxicity of combination therapies. I feel like out of this title, neural network model is the word that would be most familiar to all of you. But I do hope at the end of this presentation, you all can also be familiar with mechanistic, potency, toxicity, and combination therapies, these keywords. And the way I have created this presentation is in form of actually a journey by a PhD student. So I do hope those who are in grad school will find it quite relevant to their journey as well. And those who were in grad school will have their great sweet memories refreshed. So just since it's a journey, let's start a timeline. We are just here. We're just getting started. Diving deeper into the problem that we are trying to address, antibiotic resistance is becoming a major global public health concern. As the previous speaker mentioned about COVID-19, let's go back around 5 years, which many of us would refer to as the last normal weeks of our lives before pandemic sets in. And then these images, these things become quite normal for us. When the pandemic was there, even then the researchers, scientists, even public health experts who were discussing about next big health emergency, even when we were in a global emergency, that is antibiotic resistance. COVID-19 has claimed around 6 million lives, but antimicrobial resistance or antibiotic resistance can claim up to 10 million lives annually by 2050, which amounts to one death every 3 seconds if we take no further action. Now the question arises, like, why is this happening? Why is antibiotic resistance happening? Here is a timeline of the discovery of different classes of antibiotics. Now with time, existing treatments are becoming ineffective because bacteria, they empower themselves to fight these drugs, these antibiotics in a much better way. And evolution is obviously one of the reasons of that. And secondly, new treatments are not readily available. In the last 3 or 4 decades, we have not been able to figure out or find a novel class of antibiotics for human use. So talking about some potential solutions to this problem, combination therapy is a potential solution for this problem of antibiotic resistance. As the name suggests, combination therapy means you combine 2 or more drugs to treat a particular infection. It has its own advantages. That is, you have the optimized usage of already FDA-approved antibiotics. You don't need to discover new antibiotics. You can just use existing ones in a combination therapy, and it obviously helps in targeted antibiotic treatment. Now, there are certain issues with these therapies. There is overwhelmingly large search space. Let's say if you have 100 antibiotics to work with and you want to make a 2-drug combination out of it, 100 choose 2 will be the number of combinations you can explore from, which will definitely be an experimentally time and resource intensive process, with coupling with the fact that there is a lack of understanding of the mechanism of actions from combination therapies, and they do have toxicity and their own side effects. Now coming back to timeline, we need to keep track of where we are. Yay, we identified the problem we want to work on, but obviously now where to start to work on the problem. The first challenge which we addressed is, can machine learning be even used for drug combination therapy design, for which we did the literature review, or I should say the dreaded literature review, which was jumping from one paper to another, thinking about it's an interesting process, but in the end, yeah, it was just fine. So through literature review, we figured out that bacterial metabolism plays an important role in antibiotic resistance, which prompts us to check, can we explore integrating machine learning and bacterial metabolism information. For those of you who might not be aware of this, but metabolism is just the processes that defines the functioning of a living system. They help you process your food, your energy, store energy, and everything that's being done by the metabolic system of your body. And similar to humans, bacteria, they also have their own metabolic system. Now we talk about the approach design, how we designed it. As we know, ultimate goal for machine learning is to take some features and map them to a score or a label in our data set, that is X to Y mapping. So featureization of drug combinations, from literature, we figured out that we can have this data called omics data, which represents the quantification of impact of each individual drug on the biological system. In this case, we talk about genes. And then we integrate the data with the metabolic system, which is already available in literature, to generate what we call as the impact of each drug on the metabolic system, like which part of the system is impacted, which reaction gets impacted, or which other pathways gets impacted in that system. And then we use that information to featurize our combinations, which goes as an input to the model. And talking about the Y value, the quantification of the impact of these combinations, we use the literature data or the data from other sources, experimentally available data. So here we have figured out a potential solution. Now the task or the challenge is to getting our hands dirty with the data. So check-in challenge is how to handle data collected from different sources. Now, as I mentioned, it's an experimental data. These experiments are not done by us. They are done by other researchers as a part of prior work at different research institutes, different universities, different settings, different labs, different protocols, different people. So we are getting data from entirely different sources. So the question arises like how to handle that data. And one of the most prominent solutions is data pre-processing and normalization. Just normalize your data. The data coming from different sources, coming from different institutes, different researchers will certainly be of different distributions. So just normalize it to bring them to a uniform distribution. There are 3 different normalization techniques which are commonly used: z-score normalization, min-max, or log transform. They are on your screen. And there is no gold standard of which one is the best. It's just you try and see which one gives you best performance and just stick to it. Machine learning is more of like a hit and trial approach. You try a lot of things and you see which one hits, and then you are a machine learning researcher. Now talking about model development, training, and benchmarking, we have this omics data, we have metabolic models, we combine them together to make an input features. And as you all might be aware of neural networks, those input features just are processed in different layers to generate an output. And output in this case is a potency or a toxicity. Potency is the quantification of the impact of a combination on the bacteria or on the infection. And toxicity is the quantification of the effect of the combination on human cells. So we want high potency, that it kills the infection, but we want low toxicity, that it doesn't kill the human cells. And we benchmark our data for potency with the approaches available in literature. We saw our proposed approach is doing comparably well with these approaches in literature, and the significant result was that our approach was doing better than the literature approaches on clinical data, the drugs which were used in clinic or for human use. So yeah, hooray. We are excited. I was excited. Feeling so excited for sure. And certainly this was me and my advisor on seeing these results. Now, I won't say who is who in this case. Now, coming back to our journey timeline, we developed an accurate predictive model, right? So things should be all sailing. We should be up and onward from here. But as is the case with research, happiness does not last that long. And we performed literature review. We did that thing right. We developed an accurate machine learning model. We did that right. But as we have been hearing from other speakers in this session or in the previous session as well, scientists do not trust our machine learning model. The researchers or the clinicians, they won't trust our machine learning model. They would say like, why should we trust your predictions? Why should we trust your model? So that's the issue which we fall into. And that's a major pushback you will receive when you apply machine learning in healthcare or life sciences project in biology, where the researchers would just be resistant towards machine learning or utilization of machine learning. So now the thing is how to establish trust in your model. So here is a model design. Now talking about the issues we can have. People can say, or researchers can say, why should we trust your model predictions? You say A and B are a good drug combination. Why should we trust that? And they can say, like, why should we even trust your model design? Like the way you are making predictions, why should we trust that design itself? And I try to address these two challenges one by one, starting with model design first, because that will obviously govern model predictions. So now trusting the model design. So the solution to that is induce domain bias to the model. As we heard in the keynote session for this particular session of it, like we need domain specific tools. We just induce a domain bias to the model. It could be, if you're working on an image data, it could be your image knowledge. If you're working on a biological data, it could be your biology knowledge. If you're working on climate data, it could be your climate knowledge. Just induce that bias to the model. And how do we do that? This is our initial design. And now, as I mentioned, metabolism is a complicated system. It's a complicated process in itself. It does not just mean like one single reaction or just few reactions. There are subsections or subsystems in a metabolic system itself. So each input feature is corresponding to a particular pathway it is impacting or a particular subsystem it is impacting. So what we did, we just segregated the input features in terms of the pathways they are impacting. And let's say there are 3 pathways in the system. In real life, it would be much more than 3. And we just segregated those inputs in those 3 different sections, 3 different pathways, or 3 different input sublayers, I would say, and then process them separately as a neural network approach, and then concatenate them, and then make a final prediction. So this is what we call as mechanistic design, where we induce a knowledge which we already have from literature, from models, and just integrate that with the neural network model itself. And mechanistic design helps in better interpretation. Why so? We can interpret the weights, like pathway 2, you want to interpret the weights, or pathway 2, analyze the weights, or pathway 2. Since those weights are independent from other pathways, you can just go and start inspecting, like, why is that happening? Why a model is giving so much weight to a particular pathway than the other? And more importantly, you can knock off a particular pathway. By what I mean, you can just turn off the features for a particular pathway and then make a prediction and then compare those predictions to see if the performance has decreased or performance has increased, or if the drug combination has become more toxic or if the drug combination has become more potent by knocking off that particular pathway features we are talking about. And this is a very common approach in machine learning to understand feature importance, of comparing the model performance with feature present and with feature absent. Now we have to benchmark this mechanistic design model. We benchmarked it for the similar task as I shown before. The only disappointment or slight disappointment was that it was not doing as well as initial design, but it's still comparably well as compared to literature, and we're still doing slightly better than the clinical, slightly better on the clinical data than the literature approach, which is quite self-explanatory because there is a trade-off between model performance and interpretation. More simpler, more mechanistic, more interpretable model you will have, it will compensate that with a lower model performance. That's why you see CNNs, RNNs, large language models, chat GPTs, they are highly exceptional in terms of their performance, but when you start interpreting them, you don't get much out of it. But more importantly, if you have decision trees or random forest simplistic models, they might not be that highly performing models, but you can still interpret them really easily. Now let's talk about some of the results, drug combination landscape. Now, since we have a model which can predict potency, impact on the infection, toxicity, impact on the human cells, we can create something like a landscape where potency is on the x-axis, toxicity on the y-axis. We can do this for E. coli, the organism of our interest, one of the organisms of our interest. And we can similarly do it for tuberculosis. Both the models used to plot a scatter plot where each point is a drug combination from our whole search space. And then by convention, for both the scores, the lower the better they are. Just take my word for it, the lower the better. Lower potency score, more potent, that's what we want. A low toxicity score, less toxic, that's what we want. And then we can just figure out or single out some of the combinations which fall on low potency and low toxicity side or the section of this plot. And then in this way, we have reduced our search space from hundreds of combinations to just tens of combinations, which we can further experimentally test and move forward with. And mechanistic design reveals biological insights. That's what I was mentioning about. We have to interpret the model. Now we analyze the weights, we analyze the change in performance and in the directionality of the prediction. So here's a figure out there. It might be difficult to read for you all, but I can just highlight the interesting ones. On the top is the nucleotide salvage pathway, which shows the highest positive weights for toxicity. At the bottom is alternate carbon metabolism, which shows the lowest negative weights for potency as well as toxicity. So these findings, they sort of correlate with what we have in literature. And if we knock off nucleotide salvage pathway, it decreases the toxicity performance by 6.5%. And if we knock off the carbon metabolism pathway, it decreases the potency model performance by around 11.5%, showing that these features or these pathway features are really important for model to make its prediction or make its accurate prediction. On the other hand, if we knock off nucleotide salvage pathway, it increases the toxicity score in 88% of the combinations. That means if the pathway is not present, your drug combination is going to be more toxic. And alternate carbon metabolism, if we knock it off, it decreases the potency score in around 85% of the combinations. That means that if the pathway is not present in the model, your potency score is going to decrease. It means your combination is going more and more potent. So, update on journey timeline. We have enhanced the model design, so we are quite excited about it. Now, addressing the challenge 3B, trusting model predictions. The only way we can trust them is to validate or confirm them using lab experiments. And I'm a computational researcher, I'm a machine learning researcher, so I don't know nothing about lab experiments, so we just collaborated for it with Center for Chemical Genomics, which is an institute based in LSI here at Michigan. So I collaborated with Aaron, who has a CCG, and Kat, who is my lab mate. So we collaborated to create an experimental assay to evaluate the combinations we have predicted through our computational model. When these discussions were going on, this was me. I was not even able to figure out what's going on, just because I am not an experimental person. Or in a better visual way, this was the meeting which is going on. Now, I will just leave it up to you to decide who is me and who are Kat and Aaron in this picture. So we just tested those combinations in experiments. This is data which we got from those experiments done on kidney and liver cells, because those organs are the ones which are mostly impacted by antibiotics. We tested these drug combinations. Again, by convention, lower the score, better it would be. Just take my word for it. Isoniazid trimethoprim combination is a potent combination, and it's also less toxic. See, lower score. It's not impacting kidney and liver cells. Vancomycin is a drug which is known for its renal toxicity. Clinicians know that, like, it's gonna impact your kidney, it's gonna impact your kidney cells. So that observation makes sense with the trimethoprim because it's a positive score. Trimethoprim vancomycin combination has a positive score on kidney but not on liver. So that makes sense because vancomycin is known for its kidney toxicity. But the interesting part is azithromycin in combination with vancomycin is having a highly negative score on kidney. That is not impacting kidney at all. So that could mean that azithromycin is reversing the kidney toxicity of vancomycin, which we confirm by analyzing electronic health record data, which showed that the kidney toxicity or the percentage chances of causing a kidney toxicity with vancomycin azithromycin combination is significantly lower than vancomycin alone, but that was not the case with vancomycin and trimethoprim combination. So it suggests that azithromycin may mitigate the renal toxicity of vancomycin. Now, summing it up, the lessons we learned. Domain expertise is really important when you are trying to use machine learning for different applications. You need to be aware of what's the domain knowledge, domain bias in there, irrespective of the field we are talking about. Data preprocessing and normalization, it might be the least significant of the lessons learned during this process, but we need to, and I would definitely say, like, this could be a game changer in your machine learning pipeline. Like, your model could just not perform well just because you are not processing or normalizing your data really well. Model interpretation and model validation will go hand in hand. You need to have a model which can be pretty much interpreted and also increase the trust and reliability using validation itself. Because once a wise person said, everyone trusts an experiment except the person who did it. No one trusts a model except the person who built it. So we as computational researchers or machine learning researchers are always on the downside of this, that nobody trusts our model irrespective of the time, effort, energy we put in. Everybody's like, why should we trust you? OpenAI developed ChatGPT and people are asking, why should we trust ChatGPT? Which is a valid question for sure, but again, this is what we are in the reality of this world. Need to complete Germany timeline. We enhance the trust, reliability, model design, and prediction. That definitely asks for some kind of celebrations, for sure, right? But this is not the end. We have to think about the future as well, future directions, conduct further experimental testing, to conduct in vivo studies for more validation, better understanding of the combination predictions or the prediction of the model, to explore another way to featureize drug combinations, maybe utilize protein network information along with metabolic information, and expand it to or extend it to a wider range of therapies, maybe other infectious disease like fungal infections, which is also becoming a huge cause of concern, or even where drug resistance is a common concern like cancer. So yeah, at the end, for the acknowledgement, I will just acknowledge my lab members, Shriram, my PI, who has been constant support for me, Maggie, Rupa, Carolina, Kat for their help in this project, specifically Carolina because she was there to help when I was getting started on this project. And my friends, some of them who are in the audience right now, just helping me maintain my work-life balance, which is quite needed in grad school. And here is a QR code for our lab website in case you are interested to know more about our research. I've also put my contact info and Shriram's info in case you want to learn or chat more about our research, or in case you feel like there is a potential collaborative opportunities, do just reach out to us and we'll be happy to chat. So yeah, thank you very much for being here and questions.
Thank you for this very interesting or important application to a difficult problem. So the question I have, and this relates to your initial comment already discussed several times, that the solutions we find depend a lot on our mindset and context in which we ask them. And in your situation, the ground truth that you used was that, I presume, in cultured bacteria and cultured cells, potency and toxicity. The predictions you made in clinical applicability were rather, well, not so good. Do you have a sense for why that is, and whether there are data which can be used for machine learning or clinical applicability and potency?
Yeah, so if I get the question right, you are asking about the ground truth data which we used for the models and how to expand that to clinical applications?
Well, basically what you showed is that you can predict in vitro culture, but those don't actually give us much product.
Yeah. Yeah. So for potency models, we did use the cell culturing experiments, the in vitro data we get from the experiments, and then we put that in the model to make predictions. And I do agree with your point, like in vitro data might not give a big sense of the clinical aspect, but the toxicity model, that data is trained, or that model is trained from the data from FDA, because FDA actually has this huge repository wherein they have mapped drug combinations with the side effects they are causing, and we can just quantify the amount of side effects that particular drug combination is causing, and then we have used that information to train our models. So we assume that the toxicity model, since it's trained on the real world data or FDA data, will definitely have a better clinical relevance than the potency model data. But again, as I mentioned, we do need to again validate the predictions, and see if in silico model trained on in vitro data can actually replicate to in vivo data. So yeah. Did I answer your question? So.
It is difficult because the relevance of the in vitro culture data for clinical situations is not so good.
Yeah. That is why, like, what we hypothesize is, like, just don't select one combination out of your results, select top 10, top 15, top 20 combinations, and then see one by one, which is actually making more sense. And that's what we did with our experimental testing. Like, we did multiple combination testing, and same we can do for in vivo. And that is something which we are planning to do coming in future, to just check in vivo studies, because one of the main concerns we have is, like, even if we say drug A and drug B will interact really well, or they are shown to interacting really well in vitro, but it might not be the case in vivo, because you have to make sure both the drugs reach the infection site at the same time, which is the case in vitro studies, because you're putting at the same time in the test tube. But we again have to figure it out, like if they are applicable or relevant in vivo studies as well. So yeah.
Thank you for your wonderful lecture. In your slides, you mentioned in order to verify your model, you will do the experiment, and in your slides, it seems like 3, 4 well plates to get the data, to collect the data to verify your models, right? So my question is like, I just wonder, how can you deal with the noise, like error, and also kind of reproducibility issues when you kind of either build or verify your model?
Okay, so you are asking about the experimental design and how you ensure it's reproducible?
I mean, just like how you can kind of deal with the data, especially for the wet lab data. It's more like the errors and the noise on your data.
Yeah, so as I mentioned, I may not be the best person to answer that, but I do remember from our design, we did a lot of replicates. I think for both kidney and liver cells, we did like 12 replicates, if I'm correct, like 4 replicates on different days. So we had like 12 data points to find like one score or one quantifiable value. So we assume or we believe that with more and more data points which we have sort of reduced the noise or reduced the bias in the prediction itself. And for the experimental design, I think it was 4x4, 4x5 well-played design in this cell viability assays.
Okay, thank you so much.
Yeah, thank you very much.